This video series follows my own human bioscience lectures directly and acts as condensed notes, both for myself and for anyone wanting a head start. I hope you enjoy. The first point to make is the variety of planes with which you can cut a tissue in dissection. First is transverse, where you cut the tissue horizontally, then oblique, where you cut the tissue at an angle, and finally mid-sagittal, cut directly down vertically, stemming from the idea of the sagittal suture, a dense connective tissue joint between the two parietal bones of the skull. The tissues you are dissecting need to be prepared beforehand to ensure they do not decay. This can be done with fixatives, for instance ethanol or formaldehyde. Essentially it prevents water from being used in biological reactions, therefore preventing foreign organisms from interfering. Embedding is another useful technique, where a hard material such as wax is introduced to the cells, entombing them. This prevents movement, increasing the strength and hardness of the tissue, which therefore allows more fine slicing to take place. Now we move on to some more biological facts. When a tissue loses blood supply, for instance due to a blood clot preventing blood flow, it is called ischemia. Ischemia will cause the death of affected cells and can even have impacts on organs and tissues far away from the damaged site. This is called embolism. As a symptom of ischemia, affected tissues appear to become paler or more yellow. Finally, the holes often seen in the middle of the brain are in fact ventricles filled with fluid. In addition, on the outer layer of the brain, the folds are known as gyri while the ridges are known as suci.